Good evening, everyone. I'm Hayley. I'm the host for this evening. Welcome to tonight's webinar from Ideas to Impact, how we're going to rewrite the script on aggressive prostate cancer. We're just going to give it a few moments to allow people to join. So make sure you've got your cup of tea, you've got your slippers on, you're nice and comfy, because we've got a really good webinar coming up in the next hour or so. So like I said, we'll just give it a few more minutes, let people join, and then we'll get going. So the numbers, we're up to just over 200 people. So thank you for joining us. Just while we're waiting for everyone to log in, just let you know this is being recorded. As attendees to this Zoom webinar, we can't see or hear you. So please do get comfy, settle in. Hopefully we've got a fantastic, well, I know we've got a fantastic webinar lined up for you in the next hour or so. Just waiting on the green light from HQ to get going. We're up to 225 people now, 226. Over 230 now, the number's still climbing, so we'll give it a few more moments. Welcome if you've just joined us. We're just waiting for a few more people to come in and then we'll get going. Brilliant. I've just got the green light to get going, so off we go. So welcome to From Ideas to Impact, our webinar, where we're going to rewrite aggressive prostate cancer. Thank you for joining us this evening. This webinar is being recorded, but as attendees, we cannot see or hear you. So get comfy, get settled in. We've got a fantastic webinar lined up for you. As we go through the evening, if you have any questions, please use the Q&A box to pop your questions to the panel. Please don't share any personal health information. We do have a fantastic specialist nurse service. If there is anything personal to you that you would like to discuss, and we'll pop some information about that into the chat. Also tonight, we might be talking about some sensitive topics, including people's personal stories of prostate cancer. So if you feel that you need to step away at any point, please do step away. Please don't feel like you have to power through. Step away, get some air, breathe, and then come back and join us when you feel ready to do so. So before we get started, we're just going to launch our first poll, and we're just going to find out, if that's okay, who's joining us this evening. There's one question, and we just want to gain a little bit of insight into who's joining us, so we can get a better idea of who the audience is. This will be anonymous. We're not going to share the results. Fab. While you're completing that, I'll just run through the agenda for this evening. So we're going to hear from a lovely couple, John and Sharon Flanagan, husband and wife, who are going to be sharing their story and real life experiences of prostate cancer. Then we're going to hear from three of our fantastic researchers who are based at the University of Newcastle, who will be sharing their research on how they're planning to slow aggressive prostate cancer. And then we'll have around 20 to 25 minutes at the end to finish with a live Q&A session. So please make sure that those questions keep coming into the Q&A box. So are we good with the poll? Are we done? Fab. Thank you. So let's get cracking. So I'm really pleased to introduce you to John and Sharon Flanagan. Welcome. Are you able to pop yourself off of mute? Thank you. So thank you for joining us this evening. And I wondered if we can just briefly introduce yourself, and then John, if we can hand over to you to talk us through your experience.
Yes, certainly. Thank you, Hayley. Yeah, name's John Flanagan, and I currently have prostate cancer. I think the first important point to note is that for my personal cancer, there have been no symptoms. So I've had no symptoms whatsoever, and I received my diagnosis purely by chance, and it was following a BBC breakfast time showing of the Bill Turnbull documentary. I just happened to see that. Following it, thought, well, I've never been tested, so I contacted my GP and arranged for a PSA test. That was done, and I was called. The blood test was taken. I was called back into the surgery, and then in a 10-minute GP interview, he told me that my PSA was elevated. He did a physical check and then confirmed that I had prostate cancer. Now, I'd gone in there thinking nothing of the sort. I didn't even fully understand what prostate cancer was. But there it was. Within that 10-minute interview, I was told I'd got prostate cancer. And at that point, things sort of went into slow motion for me. But I'm currently 2 years down the line from that diagnosis, but I had a number of scans done following that. I had a biopsy done, and then I was told that I had a stage 4 cancer, that it had metastasized out into my spine and pelvis. It was aggressive, it was incurable, and it was inoperable. So all of those negative words.
Yeah.
I know, Hayley. So I then and Sharon, we had to absorb that information, and I'm currently 2 years down the line from that now, exactly 2 years. But I've been involved in 2 programs of chemotherapy, and I'm currently 17 cycles in on a 19 cycle process.
Wow.
That I finish on the 14th of next month, and I cannot wait.
You cannot wait.
Yeah. So that's where I am at the moment. And that diagnosis and that information initially was very, very difficult to take in and to process. And, you know, using words like incurable, my life seemed to stall at that moment. My thoughts started to focus on end-of-life issues. How do I tell my family? How do I prepare for it? And there were some very sad days in summer 2023. But the oncology team at Bristol explained to me that it is possible to manage this condition. And they used positive phrases like, it's something that you can learn to live with, not necessarily die with.
Yeah.
And it's something that you can die with, but not necessarily of. And they then set out a plan for me, which turned my head into positive thoughts, which I would say, Hayley, is where I am now.
Good.
Because for me, every day is a good day. And that's my approach. It might be a bit short-termism, but each day is a good day. I've said to the oncologist, I don't want to know what your prognosis is. I'll deal with this one day at a time. And that's where I'm at with it now.
Thank you for sharing, John. That's really kind of you to share so openly and honestly. I might come back to you in a moment with a few questions, but I wonder, Sharon, if we can come to you and just ask you, you know, how did it feel when you heard of John's diagnosis?
Well, initially, I was extremely angry. I really couldn't accept that a GP was able to diagnose prostate cancer correctly, purely from a blood test and a physical examination. I felt that John shouldn't have been given such bad news on the spot. And in my view, surely there would need to be some sort of biopsy to confirm it. The very idea that he was alone when such devastating news was given to him actually made me boil. I should have been with him. But by the time John actually told me, he was terribly upset and confused for the first time in his life. And for no, I'm sorry, he was upset and confused. And for the first time in his life, and for no explicable reason, a few weeks earlier, he'd started to suffer with anxiety and panic attacks. It was all so out of character, and I was already struggling to deal with this. Looking back, I believe these attacks were as a result of the undiagnosed cancer, and it was as if his body was trying to tell him something was wrong. Our lives up to that point had been so simple. We had a lovely life, lots of friends, our children were grown up, they were happy, we had no worries, we were free and looking forward to retirement. But then all of this happened and everything changed. I really didn't know what to do. I couldn't think straight. We were both in complete shock and totally ignorant about prostate cancer. In desperation, I started scouring the internet for any bits of information that might support the idea that the GP could be wrong. I found plenty. He actually wasn't wrong. But I read all sorts of reports, and in an attempt to keep everything on a positive track, I kept feeding glimmers of hope to John, hanging on to the idea that somehow it was all a mistake. John listened to me, but I knew he wasn't allowing himself to believe any of it. A couple of weeks later, when the full diagnosis was delivered, I was forced to face reality. I wasn't prepared for the news to be as bad as it was. Neither of us were. It was a lot to take in, and it took a long time for it to find the right place in my mind. No one could really help me with this. I had no idea what the future looked like. But once we'd given the news to the family and we got over the initial shock, they stepped right up to the plate. And emotionally, we are where we are today because of them. The diagnosis, together with the effects of some of the treatment, has changed our lives completely. Secretly, I continue to believe that miracles happen. It's my way of coping. It keeps my head up. I constantly watch the media for any new developments in research and remain hopeful that John's current treatment will work for a good while, during which time something new will become available for him. We've never looked for any sort of time frame on this situation, nor will we. We both try to focus positively and remain hopeful to have many good years ahead. I feel so lucky to have had 43 years married to John, and so grateful for the unquestionable love and support we've received from our dear friends and family. But it doesn't stop me feeling cheated out of the retirement we'd planned. I do try to plan ahead to give both of us little events and trips to look forward to. It keeps us going. It's also extremely important to speak openly and honestly to each other about how we're feeling. It's great to have good days, but it's also quite normal to have the not so good days too.
Absolutely. Thank you. Thank you both so much for sharing your story with us. And thank you for being so open and honest. I wonder, John, if I could go back to you and ask you a little bit more about what kind of emotions, how are you feeling when you had that first conversation with the GP? And you kind of touched upon it, but like the language that was used. And I guess what I'm kind of really keen to know is kind of how did you turn that around and turn it from a negative into a positive?
I think it took me a little while. Initially, I was confused because it was literally a 10-minute GP interview. And his exact words were, and they're still ringing in my ear, I strongly suspect that you have prostate cancer. And I'd said to him, you suspect? And he just looked at me and said, I know. and that was it. And I walked out of the surgery. There is a point, though, when I went through the negatives of thinking this is about end of life situations, but having had conversations with specialists, oncologists, and urologists, etc., who were saying, no, we can put a plan together. We can deal with this. And everybody's cancer is different. I then began to accept this was my cancer, and therefore this is my way of dealing with it. And I think once I reached that point, and I, if you like, figuratively turned in to face it, as if to say, right, okay, what have I got to do? It's incurable, but I want this to go on for as long as I possibly can. What have I got to do? So when they suggest chemotherapy, okay, and I do the first 9 cycles of it, I get a 3-month gap, and then somebody pressed the panic button and said your PSA is elevated again, your ALP reading as well is elevated, but I still feel positive at that point. And they say we need to do another 10 sessions of chemotherapy. And my view is, well, every day's a good day. Come on, let's go again.
Okay, let's bring it on.
Let's bring it on. And that's where it was. How once I'd faced into it psychologically, that began to turn positive for me.
Thank you for sharing. We have to move on now because of time. I could chat to you all evening, but we do have some interesting research to listen to. So we're excited to hear about that. John and Sharon, are you okay to come back for the question and answer session at the end? And maybe we can ask you a few more questions. Is that okay?
Absolutely, yes, certainly.
Yeah.
Brilliant. Well, thank you ever so much. So we are going to move on now, but before I do, I just want to share a few mechanisms that we have as an organisation to support anyone who's living with prostate cancer or living with someone with prostate cancer at the moment. So I think there's a link that's about to go into the chat. One of our service, obviously one we're very proud of, is our specialist nurses. So if anyone has any personal questions about they would like to talk about, whether it's from their own personal risks or the way through to I've been diagnosed, I don't know which treatment to choose, the nurses are there to help and support you. The nurses aren't just there for men either, they're also there for the wives and family of men that have prostate cancer. So please consider using that service. Other services we have are our one-to-one support service, where you can talk to a peer, to somebody who's been in the situation you're in, and you can have a very frank and open conversation about potential choices or how you feel or the emotional roller coaster that you're going through, because the peer would have been in a very similar situation, and we'll be able to help you and support you. So that's another option. We also have a partnership with an organisation called Tackle, who run local support groups. So please have a look for that if meeting in person is something that appeals to you. And we also have a wellbeing hub on the website as well. So that gives you lots of information on how to look after and maintain your wellbeing. So they might not all appeal to you, but use it as a bit of a pick and mix. Take what works for you and leave what doesn't. But hopefully there's something on there that might help. So with that, we're going to move on now to the researchers, and I'm not going to introduce them because I've seen the slides and they do a great job of introducing themselves. So I will hand over to the first speaker, who's Dr. Jennifer Munkley. So over to you, Jenny.
Thank you, Hayley, and thank you to John and Sharon for sharing your story. It's fantastic to have so many people online to hear about our prostate cancer research. Joining the call this evening, we have Professor David Elliott, Dr. Caroline Dalgliesh, and myself, Dr. Jennifer Munkley. David is a professor of genetics. Caroline is a postdoctoral researcher in the lab, and I am a principal research associate. And we all work at Newcastle University in the Centre for Cancer, and our research has been supported by Prostate Cancer UK for more than 10 years. So this evening we're going to tell you about one of our current projects where we're aiming to rewrite the script on aggressive prostate cancer. So we're going to give you some background to our research and tell you about what we're trying to do and ultimately how we hope that this will benefit men in the future. So one of the focuses of our research has been trying to understand why some prostate cancers are pussycats. So these are slow-growing cancers that are contained within the prostate and they don't necessarily need treatment, whereas other prostate cancers are more dangerous or aggressive, and these are the tigers. And these prostate cancers grow and spread very quickly and they need urgent treatment. If we can understand why some prostate cancers are more aggressive, this could be the first crucial step to try to develop strategies to stop these prostate cancers progressing and ultimately try to find some cures. And part of the answer to this is we know that genes in the aggressive prostate cancers change. So genes are like instruction manuals which tell our cells what to do. The DNA sequence of our gene is copied into a messenger molecule, and this is known as RNA, and then it's decoded to build a protein. And the DNA instructions of our genes can be read in different ways, and this is a process known as splicing. And this makes sure that our cells make the right proteins in the right amounts at the right time. However, in prostate cancer, cells can follow different instructions, and this can help the cancer grow and spread. So to give some more of an explanation, our genes need DNA. Our genes have DNA instructions, but this needs to be spliced together to make sense. So as an example, this genetic instruction has been spliced together to read, I am a cat, but a slight change in the splicing and we can instead get a tiger. And we know that the growth of prostate cancer is mostly dependent upon initial diagnosis on the male hormone testosterone. So the first line of treatment for advanced disease would be hormone therapy. And it's known that in prostate cancer, changes to genes and how they're spliced together can be dependent on testosterone. So going back to the start of our story, we wanted to understand in detail how testosterone can change the script of prostate cancer. So to do this, we grew some prostate cancer cells in the lab. So these were cells grown in flasks, and we grew them with or without testosterone. And alongside this, we took samples from 7 men before and after they had hormone therapy. And with the samples from these experiments, we used an advanced technique called RNA sequencing to sequence the samples. And this gave us a huge amount of information. And from that, we made an unexpected discovery that testosterone was changing the sugars that coat prostate cancer cells. And this led to a new area of research for me. And I started my own research group in this area to exploit these sugars to improve diagnosis and treatment. But excitingly, we also found that testosterone was controlling the expression of 2 proteins, and these are known as ESRP1 and ESRP2, and these are master regulators of splicing in prostate cancer. And they're found at very high levels in men with aggressive disease. So in our new project, we're working to understand how ESRP1 and ESRP2 change the script of prostate cancer. So how do they change how the DNA information is read? And ultimately, if we can understand that, we can work to block this process to develop new therapies. So I'm going to hand over to Caroline now, who's going to give you some more information about some experiments that are going on in the lab.
Thank you, Jenny. So I am carrying on the work that Jenny started using ESRP proteins. Around about the same time that Jenny made her discovery that ESRP levels were associated with worse outcomes for prostate cancer, there was also another group in Denmark who had a similar finding. And thanks to the new prostate cancer funding now that we've got, we're actually working together as a team. So hopefully we're going to improve the speed at which we develop things. So ESRP, the title of it gives you a lot of information about what it's actually doing. But in order to unpack all that information, we're going to need a little science lesson. So the first part of the title is epithelial, and that refers to the type of cells that these proteins control. So epithelial cells are cells that grow stuck down. Splicing regulatory needs a bit more explanation. So everybody sort of knows that DNA is the instruction manual for your cells. It's where your genes are, and it's got all the information you need to run the cells. Its lesser known accomplice, RNA, delivers these messages to the cells. And when it does this, it takes the words out of the sentences the way sort of Jenny showed you previously. But in order to get the right message, the splicing regulatory machinery is kind of like a set of scissors. And this protein here regulates how the scissors work. So depending on how the scissors are controlled, we could get from the same string of words a benign cancer with I like people, which is my pussycat, or I like to eat people, the tiger, which is the aggressive cancer. And these two messages both come from the same gene, and which message gets delivered into the cell is very much controlled by the splicing regulatory protein. Next slide, please, Jenny. So what I'm doing in the lab at the moment is I am growing prostate cancer cells in the lab. And you can see my flasks on the left of the screen there. And then there's a little image there that shows you what the cells look like down the microscope. So the cells that I'm growing have been taken from prostate tumours, and I'm growing them. And then what I'm doing is I'm modifying them. So I have made a virus, and this virus means when it infects cells, it increases the amount of ESRP1 in the cells. So it's a tool that we use in order to increase the amount of ESRP1 in the cells. And then hopefully what we can do is we can watch these cells go from pussycats to tigers and study what's happening in there. Next slide. So this is where we're at the minute. Basically, with our cells that have been infected with ESRP1, we are going through a process of RNA cell messenger spot the difference. And what we're looking for is in our cells that have higher levels of ESRP1, what messages are different to those cells that have lower levels of ESRP1. And we do this by finding the messages in the cells and spotting what's different. And then we need to work out which of these different messages could be important in terms of transforming our pussycats into tigers. So, for example, I have claws and I have sharp claws. Could both, the sharp claws make the cancer seem more aggressive and more tiger-like? So that could be a good message to target. If we could turn off the sharpness, could we slow down the cancer? The difference between I am a cute cat and I'm a cat maybe doesn't give quite as much promise as a way to tame the cancer. And my teeth are pointy and my teeth are big and pointy, well, maybe those big teeth, if we could get rid of the big teeth and just have pointy teeth, maybe that would help slow down the progression. So we need to, once we've found the differences, the next thing that we have is we need to see what effect those messages are having. And we can do that by manipulating the levels of message in the cells using other techniques. And I think that's, yeah, I think that's my section done. So the next section is going to be Professor Elliott, and he's going to talk to you about possible treatments using therapies that target the messages and see if we can use those to slow down prostate cancer. Thank you.
Thank you, Caroline, and Jenny, John, and Sharon too. I just want to follow on from the previous talks, just to say how we can use the information that we get. So what we expect to find, or what we are finding, is lots of genes which are controlled by these ESRP proteins. So the first thing we want to ask is, what do these genes do? So this will tell us exactly what kind of process the ESRP proteins are controlling. And there are data sets that we can use online, which we can also ask whether these genes have already been implicated in cancer and whether they correlate with more aggressive kinds of cancer. We can also ask questions about the actual words within the gene as well, and whether and how those modify the properties of the gene to possibly make the cancers more aggressive, and whether those words are used more in aggressive cancers. And finally, we can think about how to design drugs to remove these proteins from being expressed and remove these genes, these particular words from being used within the genes. So at the moment, we've got a particular sentence which is created by splicing, "I am a big cat," and this gives you an aggressive cancer. But could I have the next slide, please, Jenny? What we want to, sorry, one slide back, please. What we want to do is we want to be able to design splicing therapeutic drugs which block the use of a specific word within the sentence. So we're switching "I am a big cat" back into "I am a cat." And these, what we hope is using these splicing therapeutic drugs will help convert aggressive cancers back into benign cancers, which are more easy to manage. Now, these kinds of drug are not science fiction. They're actually being developed to treat other diseases. Could I have the next slide, please, Jenny? And this is the first example of one of these kinds of drugs. It's a drug called nusinersen. It's also called Spinraza, and it's a drug that does exactly what we want to do in prostate cancer. So it changes the words used by genes. And in the case of nusinersen, it's a drug that's been developed to treat a neurological disease, but these kinds of drugs are also being developed to treat cancers as well. Could I have the next slide, please, Jenny? So this slide just gives an indication of the time frame required for development of these kinds of drugs. So to give this kind of drug its more formal name, they're called antisense oligonucleotide drugs, or ASOs. And in the case of nusinersen, the first drug, prototype drug, was developed around 2000, and it did take a further 16 years to actually come out as a drug which was being used in patients and get FDA approval. So it is a long-term investment, but the experiments at the start to identify the actual target were the foundation for that, and it led on to a very successful treatment. So this treatment involved basic science, testing the drug in mice, and then phase 1, 2, and 3 clinical trials in humans as well before it got approval. Could I have the next slide, please, Jenny? And just finally, I'd like to thank Prostate Cancer UK and all the people who support Prostate Cancer UK for supporting our research and the other research which is carried on in this field. Thank you.
Thank you. Thank you all. That was really, really interesting. We've got lots of questions coming in. So just a reminder, if you've got a question that you want to ask, but you're a bit nervous, it might be a silly question. There is no such thing as a silly question. So please pop it into the chat. I'm going to ask everyone to come back and join me. And we have already had a few questions that have come through. So I just wondered actually if I could start with you, Sharon, if that's okay, and come back to you, because I kind of cut you off a little bit short because I know that you took that anger at John's diagnosis and you kind of used it in a really positive way, and I've got March for Men in my mind. So I wondered if you could tell us a bit more about what you did and how you got involved.
Oh, the March for Men.
Yeah.
It was last year. I'm not doing it this year. I did it last year and took it very seriously. And I have to say, I enjoyed every minute of it, even though the weather was absolutely dreadful. It was probably the worst March on record. It rained almost every day, but that was irrelevant. It actually gave me something really positive to focus on for the whole 31 days of March. And the preparation for it as well, because I'd sort of decided to do it about 3 weeks beforehand. And a friend of mine up in Yorkshire also decided to walk, not with me, but obviously to join the March for Men in March in support of, it was John really. That's why we did it. Our purpose really was John. And the generosity of our family and friends surpassed all of my dreams. I'd set myself a target of £250 because I'd actually done something similar a couple of years before for just a cancer research in general, and I'd managed to get 200 quid, and I thought that was brilliant.
Yeah,
the 250, it sort of moved me on a little bit. But in the end, I actually raised £1,700 or thereabout.
wow.
And our dear friend up in Yorkshire, Paul, he raised another 1,100. So,
wow.
And it was just fantastic to watch it growing every day.
Yeah.
And the comments that we received from people as they were making the donations. And it was just a fantastic experience. And John was able to walk with me a lot, weren't you?
I
did
did
2/3 of it. I would
managed
imagine.
to get a few days in, but wearing the T-shirts, bumping into men on the walk and raising awareness. And to say, Sharon was an absolute, to go back to the previous presentation, an absolute tiger with it. Every man that we crossed on the walk who asked what's the badge about or what's the T-shirt was asked, have you had your PSA checked?
Yeah. And I think what that did for us, it sort of followed on from our own decision earlier on last year to sort of come out with John's story, because a lot of men are, for some reason, very worried about talking about this. And we never were, you never were.
No,
and we've never worried about talking about it. And it's really important to talk, not just to each other, but to other people about it. And I think the more people that talk about it, obviously, it is going to raise awareness. And I think it's worked. You know, every time you put the television on now, there's a mention of prostate cancer or Prostate Cancer UK. They've been brilliant. So
well, it's thanks to people like you and great ambassadors like you getting out there, doing some fundraising, but also raising great awareness for us. So,
yeah.
So
well, I'm
thank you.
again next year anyway. So we'll do it next year.
We have a few questions that have come in for Jenny, Caroline, and David. So I'm just going to ask some of those. I've got a question for you, Jenny, and it's a brilliant question. What makes a normal body cell turn into a cancer cell?
Well, we've talked about it a bit today with the changes to the DNA instructions, but there can be lots of different factors involved. There's the changes to the DNA and the protein, but then there can also be changes to the thing that I'm working on, which is changes to the sugars that coat prostate cancer cells. And then there can be changes to other factors in the environment where the cells are growing as well. If we knew the answer to there was being one specific thing, then that would be amazing because we could change it. But we're trying to find that answer out and then reverse it.
Yeah, indeed. It's complicated, isn't it? It's not just one thing. So it's why research is really important, because we need to figure out what does go wrong so we can help put it right. Linked to your answer around the important role of sugars, how big a role does diet play on prostate cancer development in general, but also in aggressive prostate cancer?
Oh, I think that's a difficult question to answer. I think it probably does play a role, and I think there's things that people could do to help once they'd had a diagnosis. But I don't think there's like that strong a research there to say that people should be making dramatic changes. Just really being healthy and following a healthy lifestyle in general would be good to try and reduce your risk. And then, yeah, does that answer the question?
Yeah, so I wonder, when you talk about sugars, you're not really talking about the sugar that you have in your cup of tea or those kind of sugars. You're talking about kind of the sugars that the body makes, like almost the building blocks in terms of the cell energy.
So there's sugars in the diet, which everybody knows about, but then sugars also play an important role in the body, and they coat all of our cells. And that's so there isn't a clear link between what we're eating and how that changes.
Wow, thank you. Caroline, I've got a few questions for you, if that's okay. The first one is, how do you feel about working with viruses? How are there safety protocols that you use?
Yes, there are a lot of safety protocols that I use, and there have to be, but also you would think you introduce a virus to the cells, they get infected, but quite often with these experiments we have to do quite a bit of persuading to get the virus into the cells, so they're sort of not quite as dangerous as they appear, but we always have to be careful because these are viruses that can infect human cells. So yes, I follow a lot of safety guidelines, but I also spend a lot of time trying to sort of just actually get the virus into the cells, because it doesn't always just go in by itself.
That's a good answer, thank you. So a follow-up question is, how do you grow prostate cancer cells in the lab, and what fuels their growth? Which is a great question, by the way.
Yeah, so the plastic that we grow them on is a special type of plastic so that the cells stick to it. So they feel at home and they stick down. And then depending on the type of cells we're growing, we have different medias that we can use. So basically we buy them in, but sort of companies make lots of different recipes of sort of different nutrients that different cell types like to grow in. So you have to find the right mixture of nutrients to make your cells grow. But yes, the thing with cancer cells though is they want to grow. You know, they generally are sort of rapidly growing cells, so they're sort of quite amenable to being grown in a dish.
Thank you. A few questions for you, David, if that's okay. So I know you talked about the history of the drug that was that's already being used, but how long will it take for a drug for prostate cancer to be available for men?
I would imagine it would take a while. So looking back on the history of nusinersen, that was a drug that was designed and developed for a neurological disease. So it's not a cancer treatment. It's designed for a different kind of disease, and that did take 16 years. So there was a lot of research went into that during those 16 years. Probably it would take a similar time frame, unfortunately, for similar drugs to be developed for prostate cancer. Having said that, a lot of the technology which had to be developed for nusinersen was pioneering. So it involved actually a lot of chemistry, developing a lot of chemistry and delivery systems which are being used for similar drugs. So that could well speed up the the process.
So they've almost kind of laid the groundwork that others can then follow, so that it might still take a bit of time, because obviously you want to make sure it's safe and it's effective, but it might not take as long as it took them to get their drug out there.
Yes.
That's encouraging. That's
Yes.
good to hear.
It's a proof of principle.
Yeah, it works, right? Now we just need to figure out how it works for men with prostate cancer. Another one to David. Do all prostate cancers have these spelling mistakes?
Yes, that's a good question. I would say not all cancers overexpress ESRP1 and ESRP2, but the most aggressive ones do. I think all cancers probably do have some form of spelling mistake, and they also have gene expression changes compared with normal cells. So I guess the answer is yes. And they don't have single spelling mistakes. They probably have hundreds of spelling mistakes.
But I guess what's key here is that you've shown that men who have this ESRP, that's the scissors, right? So they kind of promote the spelling mistakes. They make the spelling mistakes more obvious.
Yes, they make particular spelling mistakes, which are important for the aggressive kind of prostate cancer.
Yeah, okay. Thank you. So just looking back at more questions. John, there's a question for you, if that's okay.
Yeah, fine.
There's a theme in the questions, and the theme is, what are your thoughts around a national screening programme? Do you think there should be one for men?
I absolutely do. At the time, I saw that Bill Turnbull documentary. That triggered me to ask for a PSA test. Never heard of it before, and I at the time considered myself fairly fit. I was a runner. I was running a sort of 5 miles 2 and 3 times a week. And in fact, on the morning of my diagnosis, I'd been out for a 5-mile run. Wow. So the doctor told me, I said, how do you know my PSA has risen? What are you measuring it against? He said it was tested in 2017. Now, I knew nothing about that. and he said between 2017 and now, your PSA has gone up. Now, had there been a screening service in 2017, my PSA was at 1.7. And I may well have had prostate cancer at that point, but it hadn't metastasized. It hadn't got out to the bones. And that's where my problem is now. And that's what makes it stage 4. But had I been screened when they initially did that test without telling me, then it could have been treated because it would have been contained within the prostate and it hadn't got out, it wouldn't have got out to the bones. I really think we desperately need a screening program now.
Yeah, and I agree. I don't know how much people know, but we have funded a clinical trial called Transform. And it's going to be the biggest clinical trial into screening for the last 20 years. I can see my colleagues just put a link to the trial into the chat. So we're hoping to generate the evidence to show conclusively whether or not a screening trial does help save men's lives. So we're really looking forward to that trial starting, and we're hoping to have some really positive data on that fairly soon. So thank you, John, for answering that question.
Okay.
For David, could the new drug help with advanced prostate cancer or cancer that has come back?
I think if the actual genes were selected, the good thing about these kind of anti-splicing therapies is they can be designed against any gene target. So I think if you have knowledge of the genes which are important in advanced cancer, which there is that kind of knowledge, the drugs could then be designed against those advanced targets. So the answer would be, in principle, yes.
That's exciting to hear. I have a few more questions are coming through. Keep them coming. How can we men find out about and get involved with research to help speed a cure or new treatment? I'm happy to have a go at that one if that's okay with the panel. So there's an initiative through the National Institute of Health Research, and I'm going to get the name wrong now, but it calls for people to get involved with research. So if we can find the link to that before the end of the webinar, we'll share it in the chat. If not, we'll share it when we share the recording of the webinar. But you can also look on our website. We have the clinical trials website that shows all the active trials that we are funding. And there's information on there on how to get involved as well. So it's actually called Be Part of Research, and hopefully my colleague will be able to put that into the chat so everyone can have a look at that as well. One to the panel, if that's okay. What are your thoughts regarding immunotherapy for prostate cancer? Is it challenging to find targets that are specific to prostate cancer? And I wonder, Jenny, if I can go to you first. Sorry, Jenny, you're on mute. I had to say that at least once in the webinar.
So what was the question again, Haley?
What are the current thoughts regarding immunotherapy for prostate cancer?
So at the moment, immunotherapy has only been successful for a small subset of men for prostate cancer. So that's men that have microsatellite instability. But I think there's lots of exciting research going on trying to find new targets and new ways of getting it to work.
Can you explain what microsatellite instability is?
I think it's mistakes. So it's mistakes in the DNA, but it's kind of different to the splicing that we're we've been talking about today. So it seems to, I think the trials have shown that it's worked really well for that, but not necessarily for the majority of men. But it's worked, it's been really successful for other types of cancer. But I think prostate cancer's finding some other way to hide from the immune system. So if we can find out how it's doing that, we can find new ways to make immunotherapy work. One thing that we think is important is the sugars on cells helping prostate cancers hide. So we've got that in mind to help. But I know there's lots of other prostate cancer research, Prostate Cancer UK funded research going on, looking at different ways.
Yeah, there is indeed. And if anyone is interested in reading more about other funded research that we have, you can go onto our website, and hopefully someone from the team can put that link into the chat as well. Apologies for bombarding you with links this evening. We have a question for Caroline. And the question is, would biopsy pathology have details on ESPR1 and ESPR2? So I guess would a pathologist be looking for ESPR1 or 2 currently, or is that something that's going to need to happen in the future?
I don't think it's currently being done routinely, so it's not happening as part of the diagnostic sort of screening. But we have data from sort of samples that have been taken and then passed on to researchers. So that's sort of, from that information, we know that in sort of the more aggressive cancers, we do have higher levels of these proteins. But that's not something that's currently part of the diagnostic thing. It's very much still part of the research side of things. But they're funny proteins, because if your levels are too low, because they're, if you remember on my slide, epithelial cells. So if you don't have enough of these, then your cells stop being the sticky down type. And then you can start sort of running into problems with metastasis as well. So cells need the right balance of these proteins to not be aggressive, but also not to start traveling. But yes, it's not currently part of any sort of diagnostic pathway that I'm aware of.
Okay, thank you. It sounds like it's a very delicate balancing act that you're trying to judge.
Yes, so I think that's why it's sort of better to look at the messaging downstream rather than trying to target something that's sort of potentially hard to balance.
Yeah, that makes sense. That makes sense. We're just coming to the end now. We've got a few more questions coming in, so please keep them coming. Is the prevalence of prostate cancer increasing, or is it just being diagnosed better? David, do you have any thoughts on that?
So just to check I got the question right, is the prevalence of prostate cancer increasing, or is it just being diagnosed more effectively?
Yeah, so I guess are we getting better at diagnosing men with prostate cancer? So actually the numbers are going up because we're diagnosing more men, or is there like a genuine increase in the prevalence of prostate cancer?
Probably, I would imagine we're getting better at diagnosing, and probably men are becoming more aware of looking after their prostate and knowing the symptoms and seeing their GPs. So it's probably awareness, and also of doctors as well.
Yeah, I guess it's going to be hard to pull that apart, isn't it? Because men's awareness is increasing, which is a good thing, especially when people are aware about their risks. But also we know now that for the last 2 years, prostate cancer is the most common cancer for people in the UK. So we do know there is an increase there. Fab. So I wonder if I can talking about incidence and awareness. I wonder if I can come back to you, John and Sharon, because with all the awareness you were doing on your marches, and talking to people, am I right in thinking that you actually, but a few men that did get their PSA checked and they did get go and see their GP?
Yes,
Yeah.
we do, and we know somebody who had actually got prostate cancer. So from what we were doing, from the awareness campaign we were involved in, this particular friend of a friend went and got his PSA checked, and sure enough, he got prostate cancer.
Right.
So, and we know of a number of others who have got questions raised now over it. But yeah, it definitely was effective in our little circle of people anyway, of getting men to go and get checked.
I think they all went, didn't they?
They did, yeah. We nagged them.
We did. We did. We hounded them, and certainly the majority went to get checked.
Yeah, they did.
And you know what's really important to us as well? We have a son who is 39. He's obviously sort of young at the moment, and it doesn't concern him. But he now knows about it, and he also knows that his risk is higher because of dad. And he has a little boy, so that will get passed on down to Teddy, our grandson. And our son-in-law, he's 50, he's been and got himself tested, and we've got two more grandsons. They all know.
I get a little bit annoyed when I hear people say that the PSA test is unreliable. We know it is, but it's only one indicator, and it actually starts a process.
It's the beginning, isn't it? It's the start of the process. So yeah, absolutely. So just because we've touched on risks, just to quickly run through the risks. So you quite rightly pointed out family history is a risk factor. Being a black man is also a risk factor. We know that in lifetime 1 in 8 men will get prostate cancer, but for black men that goes down to 1 in 4. And also age as well. So we recommend that men over 50 would get a PSA blood test, and that age comes down if you have either family history or you're a black man. So thank you for highlighting that for us this evening. We've got time for one more question, and the question is, if prostate cancer appears in other members of a family, does it tend to be the same level of aggressiveness, or does it depend on when you detect it? David, could I ask you to address that one?
Well, if it's appearing in the same family and a number of individuals, then it probably is caused by similar genetic factors. So you would expect that perhaps it would be a similar kind of cancer. I think another thing to look out for is the age at which it appears, because I think cancer, when it appears in younger men, is more likely to be caused by genetic effects as opposed to cancer developing in older men. So I think that's an important part of the mixture too. So if cancer's appearing in male relatives early on, then it probably, and that by early on I'm talking about less than 50, then there's possibly more chance that there will be common genetic factors.
So even more important then that they get checked earlier.
Definitely.
So yeah, absolutely. I'm going to draw it to a close there. I'd just like to really thank all of our speakers. Thank you to John and Sharon. Thank you for being so open and honest and sharing your story with us this evening. Really massively appreciate it. Thank you to David, Jenny, and to Caroline for giving up your evening and to talking us through the fascinating science that you're working on up in Newcastle. And thank you to everybody in attendance for listening and for asking your really good questions. So thank you very much. I do just have a few things to run through quickly. And that's to remind everybody to take the risk checker if you've not already taken it, or to share it with your friends, your loved ones, through whichever means, be that social media, or through your WhatsApp. And also just to highlight that the research that we fund, we don't have any support from the government to fund this. We fund all of this through people's hard work in fundraising, people like Sharon going out and doing March for Month for us. So again, another thank you to Sharon, but also a thank you to everyone who has done some amazing fundraising for us. And also an opportunity that if you'd like to support us, we're just going to pop yet another link in the chat, to give you an opportunity to help us to fund more life-saving research in the future. We're just going to launch our last and final poll, and this is a feedback poll, so give you the opportunity to tell us how you found this webinar. This will help us make sure that we maintain the high standard that we aim to achieve each time. Again, these will be anonymous, and there'll be an opportunity for you to share further feedback and ideas in a post-event survey, which will be emailed out to you tomorrow. And while you're answering that poll, just to let you know that today's recording and an FAQ, so if we didn't get to answer your question, we will attempt to answer it and send that answer around with a recording of the email at some point, hopefully next week. So thank you again for everyone who took part in this evening's webinar, especially our speakers and also everyone at home who asked great questions. Thank you very much and have a lovely evening.